So student, today we will discuss about the rRNA and tRNA. Okay, so as you know, RNA is one of the most important genetic material which is present inside the cell, and RNA is only responsible for the protein synthesis process. Clear? But if you remember, we had discussed about the central dogma of life, when main concept of central dogma is when DNA just send its information in the form of code, means cistronic form, then only mRNA gets to be synthesized. Okay, and from this mRNA, with the help of rRNA and tRNA, polypeptide chain or protein chain can be manufactured. Okay, so the whole concept or the whole mechanism of it is only known as the central dogma of life. Clear? So already we had discussed about the mRNA. So today we will continue it with rRNA. Okay, rRNA is basically associated with ribosomes. Clear? And it's not having any specific type of diagram that by which you can understand, because RNA is generally associated, as I told you, with ribosome. Because of it only rRNA is the largest type of RNA. Okay, so and it very helpful in the production of or in the synthesis of protein. So basically the main function of rRNA is it is associated with ribosome, and it helps in the protein synthesis. Many more few terms are related with this concept, and they are very important from the point of any competitive exam. Clear? So here I am going to discuss some points which are very important. Okay, so first point is that a part of DNA, clear, a part of DNA which help in the production of protein in future, that part of DNA and part of DNA which code for a particular protein in future, that part is known as the cistron. What is known as cistron? Cistron is a one part of DNA which help in the synthesis of protein in future. Okay, so one segment of RNA which have cistron. Generally, you know, DNA can't synthesize directly. DNA has to send codes with mRNA, then only the protein can be manufactured. Okay, so one segment of RNA which have cistron, if it code for only one protein, that condition is known as the monocistronic. Are you getting my point? If RNA, a segment of RNA which have a same type of cistron, means monocistronic condition, so this will help in the manufacturing of one protein. Clear? Like this, if it synthesize more than one protein or polyprotein, that time this condition is known as the polycistronic condition. Okay, so next thing is about, next point is, what is the C value? C value means total DNA which is present in one set of chromosome. It is only known as the C value, like N or 2N. N is represents haploid condition, 2N represents diploid condition. So what is the C value of it? C value of haploid chromosome, it's 1. Okay, or C, or in case of 2N it's 2, or I can write it 2C. So this is known as the C value of DNA content. Okay, as you all know that human genome has 3 billion base pairs and the length of DNA is 2.2 meter. Clear? So you can understand by your own self that how that much long DNA is able to stay inside the nucleus. The main reason of it is packaging of DNA. Clear? Histone protein is very helpful in the packaging of DNA. So if DNA is not packaged, that time the length of DNA is 2.2 meter. It's not a very small length. Okay. So next thing, what is the mobile DNA? Mobile DNA or jumping genes. this is a very important term, children, related with this content, that mobile DNA or jumping genes, they are very important because they can change their place in genome. Okay, so here one more important thing about, generally we know that DNA is present in a double stranded form, but some exceptions are there also where DNA is present in single strand. Few example I am telling you. One example is coleophag or phi 174. Clear? They are the two viruses. Which type of viruses they are? They are group 2 viruses. Okay, so these viruses can contain single stranded DNA. Also, generally we know RNA is present. You can see on your screen also, RNA is present always in a single stranded form, but some exception is there. RNA can also be present in double stranded form. Like example is again in one virus, that is of the Rio virus. Okay, so RNA is also present in double stranded form in Rio virus. Okay, next important thing which can be asked in any competitive exam, that what is naked DNA? What is naked DNA? If in DNA histone is not present, okay, so if histone protein is not present, that time DNA is known as the naked DNA, or DNA without histone protein is known as the naked DNA. I hope these points are clear. They are very important points and you should know about them. Now I am going to tell you about tRNA. Clear? tRNA is the smallest RNA, children, and also known as the soluble RNA. Okay, so the question can be asked that what is the soluble RNA or what is the smallest RNA? And one more thing, they are also known as the adapter RNA. So the answer is tRNA. tRNA is also known as the adapter RNA, smallest RNA, and soluble RNA. Okay, and what is the largest RNA? Yes, rRNA is known as the largest RNA. Okay, so as on your screen you can see that one structure is there. If you open this structure from this side and from this side, you get a single strand. Clear? But as you know that nitrogenous bases can be make hydrogen bond. Because of this only tRNA is having one specific type of structure, and this structure is known as the clover leaf model. Clover leaf model. One question has asked in one examination, I have also heard it, that clover leaf model is generally associated with, or tRNA is having which kind of structure? Okay, so that time the answer is clover leaf model. So clover leaf model is associated with tRNA or transfer RNA or smallest RNA, soluble RNA, and adapter RNA. Okay, so in this structure you can see that different arms are there. Okay, so here this end is known as 5' end and this is 3' end, or you can see here in 3' end ACC is present. Can you see? ACC is present. Okay, so that's why it's of 3' end, or it is only having one OH group, means one functional group, and this is known as the amino acid binding site, means amino acid generally binds in this side only. Okay, so this is known as the amino acid binding site. What is the main function of tRNA? tRNA helps in the bringing of amino acid, and these amino acid come on with the help of tRNA or adapted by the tRNA, and these tRNA leave these amino acid in the form of chain. Clear? Mean one tRNA bring one amino acid, other tRNA bring other amino acid according to the coding on mRNA. Okay, so only in this sequence only, or when the all amino acids bind with each other with the help of peptide bond, then this peptide chain is only known as the protein chain. Okay, so here you can see, so different arms are here. You can see it's known as the amino acid acceptor arm. Okay, and here you can see, very simply I am going to tell you, T arm. This is known as the T arm. Okay, so what is the T arm or T loop? Here you can see that one important thing, G, T, or then you can see P, that once phi sign is there, it's known as the P, C, G. Can you able to see this structure here? Okay, so this sequence is very important, and this is only known as the pseudo uracil loop or pseudo uridine loop. Okay, so here you can understand that this loop is responsible for the recognition of ribosome. Clear? Firstly, the ribosome also has to be recognized. मेन फंक्शन ऑफ टीआरएनए का मेन काम है एमाइनो एसिड को साइटोप्लाज्म में से लाना, लेकिन लाकर उस राइबोसोम के पास ले जाना जो राइबोसोम एमआरएनए से कनेक्टेड है. ओके, तो राइबोसोम के पास ले जाना, उस राइबोसोम को पहचानना, रिकॉग्निशन ऑफ राइबोसोम कैन बी डन बाय दिस टी loop. Okay, and the down one side, this loop is known as the anticodon arm or anticodon part of loop. Okay, I can tell you this very easily. Here you can see three triplets are GAA. So this tRNA can bind on such mRNA which is having CUU code. Okay, CUU code means C is generally linked with G, U linked with A. If you remember, in DNA in place of U, uracil, thymine is present. Okay, so A. So this code is identity, means firstly this code is present on mRNA and this code is identified by this tRNA. According to this code only, amino acid can bind in this acceptor arm, then only the amino acid come on this area. Are you getting my point? So this anticodon arm which works, or only such tRNA can bind in on mRNA, the code is already exist. Okay, so it's about the anticodon arm. The third one arm is known as the D loop or D arm. Why it is D? D means dihydrouracil. Clear? DIHU, dihydrouracil. And the function of this D arm is it helps in amino acid recognition site. Okay, amino acid binding site is different, acceptor arm is different, but